Hello and welcome to the Spatial Transcriptomics Explorer. This is an interactive tool for exploring gene expression in the bird's eye. So if we look at the first tab here called Differential Gene Expression, we can compare the results of differential gene expression from two analysis methods. On the left, spot level analysis, and on the right, pseudobulk analysis. And the significant genes in the volcano plots are shown in red and the non-significant ones in gray. And we can actually hover the cursor over these spots to see the gene names, p-values, and the fold chains. So this is, for instance, the MMEL1 gene, and this is of course also possible for the pseudobulk data and for the gray non-significant points. And below these volcano plots we have these sliders, and these sliders adjust the thresholds for log2 fold chains and p-value. And when these are changed, the plots and above and the tables here below updates automatically. And the tables here below are showing the top significant genes. So let's see, and this will of course update when I change this slider. So if I change the log2 fold chains to 2 and maybe a very high p-value of 80, and say push apply filters, both the plots and the tables will then update. So here you can, for instance, see that there are a lot less significant points and significant genes here in pseudobulk analysis, and then of course also a lot less of the top significant genes. And then if you have a lot of genes, for instance in the spot level one, you can change the entries, so maybe you want to see all 50 at once. And then it is also possible to search or sort this table to find specific genes of interest. So for instance, if I am interested in the carbonic anhydrase 4 gene, I can search for that, and then I can see, oh, it's the number 30 on the list of top significant genes. I can see the log2 fold chains and the p-value. And lastly, if we are interested in the spatial location of a specific gene, we can also select it here. So maybe I am super interested in the ENG gene. It is actually linked to this Tissue and Embeddings tab. So this tab is, in this second tab, we can actually explore where individual genes are expressed in the eye tissue and how the data clusters in different dimensionality reductions. So this over here is the spatial tissue image of the bird's eye, and it is colored by average gene expression, as you can see here with the color gradient in the legend. And then we can actually select genes over here. So now we chose the ENG gene, so now all of these are colored by, let me just clear the selection, but all of these spots are colored by the average expression of ENG. But we can also maybe there's another gene that we're interested in, maybe STMN2, and then it changes. So then the average expression changes in all of these plots. And below we have the different dimensionality reductions. So PCA, UMAP, and TSNE. And these all show the structure of the data by grouping similar data point, and each dot is a spot from the tissue. And then we can select a group of spots in any of these 4 views. So the same selection is then highlighted across all views. And it works both ways. So for example, if we want to know whether a specific structure is its own cluster and whether this is true in all types of dimensionality reduction, we can just take this brush, this one, and we can then select this structure, for instance, in the eye, and then it would be highlighted in all of the dimensionality reductions, showing that this is, for instance, has its own specific cluster. But additionally, maybe we will also observe something looking like a cluster in one of the dimensionality reductions, and we want to see the spatial location of that. So let me just clear the selection here. Maybe I look at this part and say, this actually looks like a cluster in the UMAP. And then it would highlight in all of the other dimensionality reduction plots and also in the spatial feature